And the lecture is about single-cell RNA sequencing data analysis. I do it in R. We are going to do today it in R. A little bit about me. I'm Ukrainian. I'm from Lugansk. Then I got a medical degree. So I'm an NMD by training in Russia. And then I moved to Aarhus University, where I'm doing research on smooth muscle cells and atherosclerosis. And because smooth muscle cells are very interesting and change their phenotype and have a high level of plasticity, we became interested in single-cell RNA sequencing because it allows to understand heterogeneity in this cell type. And that's a kind of introductory basic slide, why we can be interested and what questions can be addressed with single-cell RNA sequencing. Of course, it's as a biologist or molecular biologist, tissues contain different cell types. And when you do bulk RNA sequencing, trying to understand any transcriptomics data, and then you compare your condition and case, you probably will be addressing questions in cell type composition and not transcriptomic differences in individual cell types. This is a basic application of single-cell RNA sequencing data analysis. Then we can do some sophisticated things, not really sophisticated, now it's common, like trying to understand some maybe rare populations, trying to understand cellular states in processes. It's also very important in development, embryological development, because we can actually track transcriptomic differences along various differentiation or pathological processes. And then we can use various mathematical models to trying to generalize processes that we see. Currently, there are 4 technologies to address single-cell RNA sequencing. Single-cell and single-nuclear RNA sequencing. So basically, they're divided by 4 technology groups. It's the most common is emulsion-based technique. That's 10x, and this one, PIPSEQ, not really used. Then also, we will be focusing on analysis of this type of data. Then also it's a microvel with Rhapsody. Then it's a combinatorial indexing, quite interesting actually idea, because it allows very cheaply get lots of cells sequenced. And this one is something really new, hydrogel based. And there was benchmarking on trying to understand which of these technologies is better and which you should choose, when you should choose which one. And basically, this one, combinatorial indexing, is pretty good, but you have a high amount of cells, but they are poorly sequenced, like low depths. And this one allows you to sequence with a low error rate, but then cost is higher. So you typically have a trade-off on that. There are other, you can go to this benchmarking, not benchmarking, this study that compares these different technologies. So in case when you choose application and technology in your particular study, then you can have idea what pitfalls are lying in each of these technologies. We will be focusing on 10x because most of the data nowadays are obtained from 10x technology. And basically this is your tissue. Then it can be, for single-cell RNA sequencing, it should be fresh. And for single nuclear, it also can be frozen. And actually, this combinatorial way to do single-cell RNA sequencing allows you to use paraffin-embedded tissue, which is super cool because it's a lot of archives. But anyway, you work with tissue, and tissue contains cells that are embedded in a cellular matrix, so you need to dissolve the tissue. Let me ask first, actually, because this is very basic. How many people, raise your hand, worked with single-cell RNA sequencing? Okay, I think that was 1/4, basically, maybe less. Then, yeah, we will go a little bit more in depth. Basically, you break the extracellular matrix, then this is your it's barcoded gel beads. You buy them from the manufacturer, That's your cells. And that's oil. And basically the idea that you have one bit, then you need to mix it with one cell. And this is a reaction that is going in each individual. Then this is a reaction chamber. And that's a reaction that is going in the reaction chamber. Pretty important to understand what are these gel beads. This is based on Illumina sequencing technology. So you have each gel bead has thousands, millions of such nucleotide sequences. This one is basically this one is needed for sequencing. Then you have a 10x barcode that allows. This is basically a barcode for a cell. This is UMI that allows you to find the PCR templates, and this one. Does anybody know why do we have poly T end? This is the first question. It's an opportunity to get candy. Yes. How do you fish out the RNAs? Yes. Good. That's true. So basically, then we do all these reactions that correspond to typical NGS sequencing and do the sequencing. Basically, it's again, while we are working computationally already with the data, you will always refer to number of UMIs. It's basically a number of unique molecules of mRNA. They can be multiple of the same gene, they can be different genes, so it's all mRNAs that you got from your data. And this one is the index of your cell. And I will skip all the alignment step because we actually have Maya to cover, I think, alignment, and Andrei, right? Andrei. Okay, and we will start. Imagine that you have already aligned everything. So we go back in future. So you have aligned already everything. And basically, how from the sequencing machine you get FASTQ files, and you kind of need to get to answer a question which mRNAs belong to which cell. To do this, you exactly do the alignment using some reference genome. And then, typically for 10x, you use software called Cell Ranger from 10x company. Then you have this sequence, cell barcode and UMI. And here, this part is aligned to the reference genome. You are grouping cell barcodes, grouping UMIs, trying to understand which molecules of mRNA belong to which cell. And for example, here all of them, they are unique, and then you're comparing UMIs. That's important because of such thing as PCR amplification bias. Because when you amplify mRNAs, you also amplify UMI, and so you need to understand, is it just the molecule that was just amplified, or it's a real original mRNA? So if UMI is identical, therefore it's just duplicate, and you discard it, and then you end up having you delete all the similar UMIs, identical UMIs. You count unique UMIs, and this is your final row count matrix. It describes expression of thousands of genes and thousands of cells. This is your cell barcode, and that's your gene. And then the magic, all the diversity of ML-based methods start coming to play, because basically it's just matrix, and you can do whatever you want to do. This is typical pipeline. You can refer to this very nice paper from 2023 that describes common pitfalls, approaches, how to do single-cell RNA sequencing data analysis. Typically, the pipeline contains QC, normalization, feature selection, integration, You can read it further, and we will go a little bit also here. And the whole idea of this, of my lecture, is actually trying to cover all the pitfalls and go a little bit in depth, so we know. Of course, we will cover all the mechanisms that are going inside of each this step, but also what pitfalls we need to understand to do it more robust. Why do we use CRAM? Well, first comes to play that I know R, that's why. But also because it's a highly cited and robust package. Nowadays, different benchmarks show prevalence of Seurat-based methods. Later, we will cover which parts of the pipeline of this pipeline overperform other methods. So it's a quite good tool. It's quite comprehensive in the ways what you can do. It's highly updated and developed, maintained, and you need to less struggle with various problems like incompatibility of matrices and so on and so forth. Because when you work, for example, when you switch between Python using ScanPy and R, it's not so straightforward. There are like different, how even, for example, logfold change during differential expression analysis is computed in ScanPy and in Seurat, it's different. So it's difficult to switch actually between these two frameworks, although possible, and we choose R because it's just comprehensive, and in R there are plenty of packages that allow to do some downstream analysis. Seurat names comes from the painter. They're painted with dots. So the first step is QC analysis and quality check. That's your typical, what you load to the sequencing machine. It's your GLBit, and it's just lots of garbage. Basically, you are mostly interested only in this cell, and all of this you want to discard, but then we need to to find out how computationally we can distinguish this cell option of the GLBit from all of them. So we can get clean emulsion. There was a quite interesting study because they had ground truth ground truth cells divided to categories high quality, multiple, like doublets, broken or empty, and they tried to measure all possible biological or technical variations, including like a number of aligned reads, multi-mapped reads, genes expressed, trying to find out how we can actually distinguish broken cells, dead cells, from live high quality cells. And the problem that it was not so straightforward. First of all, it highly depends on cell type. Because different cell types, for example, it's very well known that T cells, they have a lower amount of in feature and in count. And therefore, if you're just trying to introduce some very rigid thresholds, you will filter out or under-filter some populations. Depends on this, of course, thresholds. What they found that what we actually know, overrepresentation of overexpression of mitochondrial genes in a broken cell, but it's not that high. Although, of course, we can use it. But if your cells, it also, of course, depends on your protocol. If your cells are also like cell type specific and they, for some reason, under some condition, express a lot of mitochondrial genes, then it can be, of course, a problem, but you should be aware of that. And then another, what also helps to distinguish damaged cells from live cells, is number of mapped reads and multi-mapped reads. It's lower in broken cells. And then nothing else actually works. So,
sorry, is this the same for fresh frozen and for fresh tissues?
They used only fresh tissues. They didn't use fresh frozen.
Okay, but this would be different, right?
Yes. Yeah, it's highly dependent on the way we sequence. Of course, quality of the tissue. Here it's only fresh, and they also use the same protocol for all the tissues that they sequenced.
And it's possible in cellular to count this in?
To count what?
Like the medical tissue extraction we should use.
We can model if you have in the same data set and you have like a variable. You can model and include it in model, but it doesn't mean that you actually can count on that. So, yes. The result of your sequencing is highly dependent on technology that you use, pre-processing step, processing of your tissue. Enzymatic disaggregation also introduces lots of changes in expression profile. If anybody is interested to talk about wet lab and this thing, we can talk about it. Maya, do you want some candy? Okay. I can pass.
Thank you.
Another thing that we should take into account, that is actually rarely taken into account, is ambient mRNA. While we embed our cells in these gel beads, some cells are leased before they get into the bead, and therefore there is always mRNA that is just in this soup. So basically, ambient mRNA. These molecules, free molecules of mRNA, always gets into some beads, and therefore it kind of unifies all the background that you have in your cell sequenced. There are two ways how you can deal with it. One is just you trying to find the unit for that raw data from your sequencing machine. One approach is that you're trying to learn the background of empty droplets, and then you're just trying to subtract all this background from all the rest of beads. And that's how SUPX, for example, works. And then you're trying to actually also identify cells that have high amount of this background ambient mRNA, and remove all of them. Another approach is trying to modify the count matrices, subtracting this background. So these approaches work. Empty, cell bender, these approaches work in this way. There is actually no benchmarking study on ambient RNA removal, but we should be aware that if you're working with highly damaged cells, no, not highly damaged, but prone to be damaged, then you will have a lot of ambient mRNA here floating in your soup, and you will have higher mRNA background. Yes, please.
Oh, sorry, just like to ask that if you remove the background just presenting all the cells, how can we distinguish between the housekeeping and the
You are based on empty droplet, and if it's a housekeeping gene, if a cell is alive, of course, there are some housekeeping genes and thousands of them, but it's not that they are in empty droplets, so they will not be learned by the model. Which one do you want?
The right one.
Thank you for your question.
Thank you very much.
Speaking of that, how do we know if a bead is empty?
Based on the number of sequenced genes and UMIs. But of course, it's very low, typically, and that's how we assume, we don't know, that it's empty.
So it's just based on some thresholds?
Yes, yes.
Sorry, about this other approach with removals in dark code based on expression profiles. How do we know which expression profiles are to be removed? Like in the approach with the removal based on empty droplets, it's easy because you have the empty droplets. You can just see what RNA you should remove in all data set. But in this other approach, they
all start from learning background on empty droplets. You feed the raw data from Cell Ranger, unfiltered data, to these approaches. Just this one is modifying your count matrices, but trying to preserve all cell beads, like cell droplets.
It's not removing the droplets, it's just modifying the whole matrix.
This one, yes. And this one removes droplets. Next, what we need to do, so we identified and tried to find one end of this scale of your low quality cells. It's cells with a low amount of mRNA or ambient mRNA. Another scale of your problem is the doublets, and it's actually a huge problem, and we are going to dive a little bit deeper on that. Doublets, when your cells are small, or maybe you have a strong extracellular matrix that is not completely broken by enzymes while you're preparing your libraries, then cells can stick still together, and while you're preparing then emulsion, several cells can get into the bead. That leads to a problem of doublets. Doublets can be homogeneous, which means that the same cell type goes into the bead, or heterogeneous, which means opposite. There are some, because it's an important problem, there are some benchmarking studies on and several approaches to identify doublets. Doublet finder was proved to be one of the best. Then another, this one, SC double finder, overperformed it. And then because there are now actually more than 20 different approaches to identify doublets, package doublet collection was developed to kind of together all of them, and run is one command, so you can understand the output from different algorithms. The thing, for example, look at this. This is approach from SC double finder, but most of them, they work in this way, just different mathematical models. You don't know the ground truth. You have the emulsion, you sequenced it, and you don't know actually which cells, these individual cells, they are doublets. So what you do, first you cluster, and then you're, based on clustering and graphing KNN construction, you find cells that are more similar within their cluster than with other clusters. Then you just take cells from these two clusters, mix, and try to find if there are clusters then that look like mix of these two. Which already an approach that has lots of questions, because, for example, if you work with diversity that is continuum, then you take ends of two ends of your continuum, mix, and you have something in between, and actually this cell type can exist in real data. So in doublet detection, there are lots of these problems. You need to be aware of your cell types that you expect in your sample, so you do not overcorrect and do not remove real cells because they're just similar as like mix of two cell types. For example, when you run double collection, and then in our workshop we will run two different methods later. You will run two methods, SC double finder and double finder, and they have less than 30% identified the same doublets. So almost 70% identified doublets by individual methods, they do not overlap, which raises questions, of course, like to trustworthiness of these methods. Of course, it's up to you as an analyst trying to identify if you really found some doublets, or it's just a specific cell type that has higher in feature, higher in count. For example, cells with stem cell properties, they have higher diversity of genes expressed, up to 10,000, and they have a higher amount of unique mice, and therefore they can be considered as doublets. But conventionally, so to come to other variables of your cells, what else we can do to clean up our data? Well, yes, you look at the number of unique molecules expressed, identified in your doublets, and this is typically distribution that you have. You have a lower amount. Here it's your empty droplets, and you can also set some thresholds here, assuming that these cells are doublets. This is a number of detected genes. There is another parameter that is actually very good. It's called library complexity or novelty score, which is a ratio between the number of genes identified and number of mice identified, which corresponds typically if you have a doublet then you will have lower this score because you will have the same genes expressed with higher amount. So you with higher amount of UMIs, which is good sometimes. So sometimes it helps. There was another metric developed to identify bad quality cells, which is called mito-ribo ratio. From biology, we know that when cells are alive, they have some ribosomal genes. They are vital ribosomal protein genes. And they have a lower amount of mitochondrial gene expressed. So ratio between these two helps to identify 3 categories of cells: live cells, pre-apoptotic cells, and apoptotic cells. That's computationally and not completely benchmarked, but possible to use if you are interested in deleting dead cells. Key takeaways from quality control. So try to plot all possible combinations of all the QC metrics, like feature and count, number of mitochondrial genes, number of GAPDH, all the glycolytic genes. So you can identify bad quality cells that are not completely alive. You need to know your cell type because all these parameters, like number of, like glycolytic activity of your cell type, depends on the, of course, cell type. And if you find some outliers, you should consider removing it, probably. You can set thresholds based on your own knowledge of the cell type or based on some, for example, statistics like MET. Yes, we need to think about doublets and how we detect them, but we need to do it consciously. Doublet detection, they are learning model of doublets on individual data sets, so you don't need to use it on aggregated data. Then try to always do PCA analysis and explore PCs and loadings on PCs, so you find some confounders, hidden confounders in your data. And try to retain low quality cells so you can actually always go back and understand what these bad quality cells are. But if you still retain low quality cells in your data set without identification that they are low quality cells, it will drastically change your subsequent analysis, so be careful. Because that study that I was referring to, that was studying broken cells, they found that retaining low quality cells changed differential expression analysis, like 70% of genes, they were differently expressed in comparison if you remove the low quality cells. Next step is normalization. Do you have, let's stop here. I will take a breath and you will ask probably questions. If not, then I will just take a breath. Yes, please.
Can we rely on percent of metrics to detect empty cells instead of using such sophisticated models like you discussed?
Well, typically you discard empty droplets just based on number of identified UMIs or features. There are no like really sophisticated things. You just remove everything that has like less than 200 UMIs or features expressed, and that's all. But number of aligned reads you can't use completely for this purpose because some healthy cells, they also have low this low number, and there is no any metric that as a single metric help you to identify low quality cells.
Oh, and about the opposite case, when we try to check the tablets, I suppose we also can estimate it by percent of reads.
Yes, we can, but also it's the same. Like you use it as a composite. Loading into your brain, trying to identify what is the profile of unwanted cells. Did I answer your question?
Yes, thank you.
Yes. Oh, sorry, yes.
I have a question about if chromosomal silencing in female mammal cells. Can it be helpful for finding doublets, or the same X chromosome is silenced in the same small tissue sample?
On silencing, but if your X chromosome is silent, how would you know the expression profile?
I mean, that only one copy of X chromosome with expression active, and I wonder how how small is the scale of this mosaic? Like, can the allelic diversity of X chromosome genes be useful in finding doublets, or it's the same X chromosome active in the tissue sample?
The problem that while you're using most of the technologies, you sequence 19 nucleotides. So you can't really understand allelic things, or you can't really address any genomic question because you don't have enough data to do that.
Even if there's only work in females, right?
Yes, but
It would be interesting study, I agree. It would be very interesting.
Probability only that they are different, even if they're inactivated totally random.
There is allelic imbalance. So if you use packages like allelic, you can calculate the preference of if a gene comes from one copy or the other, and that can also be diagnostic of maybe one of the chromosomes not being switched off correctly. So if you have like 50-50, you shouldn't have 50-50.
Yeah.
Because one chromosome needs to be completely activated, so you need all of the genes coming from one chromosome.
Did you have also a question?
Yes. As far as I understand, both the UMI and the barcode are random nucleotide sequences, right?
Randomly what?
Random nucleotide sequences.
Yes.
And why do we need like both? Aren't their functions kind of...
No, they're not overlapping. Barcode is unique for cell, and UMIs can be the same in different gel bits, and UMIs help to identify this PCR bias, PCR duplicates. So UMI is needed for identification of PCR doublets,
Yes.
and the cell barcode is for cells.
Genotype, so the barcode needs to be the same for the whole droplet?
Yes, the barcode is the same for the whole droplet.
And the UMI is totally unique?
Yes,
Okay.
for in wild gel bit, it's unique.
I have a technical question about.
Yes.
So, in your opinion, what is better? To set, to like look at the sample, set some strict thresholds, like 200 features, for example, upper, lower threshold, and so on. Or if I'm lazy and I want to use this package from bioconductors that just add per cell QC metrics and then just based on MED labels the cells, and sometimes for some samples the thresholds look like they look different for different samples, but it looks okay. Like, is it okay to just use this MED approach and not bother with setting the threshold manually?
MED is trying to find outliers. And the problem with outliers, if you have in your sample, if you have a bunch of bad cells, then they won't be outliers. They won't be pretty much a decent part of your sample. You won't find them as outliers. Does it make sense?
Yes. So, but if I know that I did the cell counter synth before loading the cells into Chromium, and I know that I have like 90% viability in the sample, then MED approach will be relevant.
Low quality cells, it's not only dead cells, but it's also doublets. And the vitality of your sample doesn't mean that you don't have doublets.
Yeah.
So you still need, the key is to understand your cell type.
May I ask you a question?
Yes.
So one thing is that, okay, you have PCR duplicates and so on. So one thing that when you work with the hashtagging of the cells and so on, this is the problem that people don't seem to address quite often, is the PCR swappers, right? So you have reads, but they're actually they're very similar in terms of sequence. So then you have polymerase that jumps from one template to another. And what it happens, at least when we try to hash cells, is that the cells, the big fat cells that have a lot of reads, they kind of swallow the smaller cells. And this is something that you also expect to see in normal data. Let's say I have a PBMC data set, I have erythrocytes synthesizing hemoglobin, and since I have so much hemoglobin that it takes over the barcodes of other cells. So I wonder if you ever analyzed cells and you look exactly at the genes that they express, for instance, macrophages, do you see expression of erythrocyte genes? Do people assume it's an ambient RNA, or because it should be that the big, highly expressed genes take over the barcodes of cells that are not, in fact, expressing? Have you ever seen this?
Yes, I haven't worked with this question, but in my data mostly, the problem that macrophages are fragile, and the big cell types typically are fragile, so you typically have a strange mix of two cell types, but it's not doublets because they don't have higher amount of the feature. There is no any approach to address this question. I haven't found at least anything.
So you do see cells, the expression of profile which doesn't make sense, but they're not doublets. It's just contaminated.
Yes, and I was speaking to people that work with spatial transcriptomics, and they said that it's in spatial data it's common because you have this problem with cell segmentation, and there you actually can find the transcriptional profile that correspond to transcriptional profile of this strange cell that you find in single-cell RNA sequences, because I think it's pieces, their hypothesis was that the pieces of cytoplasm go together, and cytoplasm doesn't contain a lot of, so you still have a normal-looking cell, but with weird expression profile. But it's not proved, and I think it's something that will come later, maybe. It's just... based on experience, how you see your cell type, I think that this question is addressed, right?
If you know what you're looking for, if you know your cell types, you can say this doesn't make sense.
Exactly. That just doesn't make sense. But some people will disagree because in some fields, people still think that there are smooth muscle cells that acquire macrophage properties, which just doesn't make sense at all. But because in single-cell RNA-seq data, they believe in it. It's a huge problem in the field.
You can validate.
The problem that, it can be, we can discuss it later. Let's continue to normalization. Okay, you have cleaned up your count matrix. Then you have row counts. And now I have next question. What is the distribution of single-cell RNA sequencing data?
Negative binomial?
Yes. Negative binomial, who else said what? Which person?
Okay, let's say
Yes, gamma Poisson. Poisson.
Yeah.
Yes, that's true. Knowledge and distribution, we need to understand how we can address problem with normalization. And another thing about single-cell RNA sequencing data is it's heteroskedastic. That means that while you are having difference between 1 and 2, this is much more relevant than having difference between 100 and 101, which is heteroskedasticity. So we need to stabilize variance. Another thing why we need normalization, it's depicted here, because if you have lowly expressed genes, for example, transcription factor, that you detected in one copy, in one cell type, and in 2, then the difference will be 1. And here, because it's highly expressed gene, then the difference will be much more. But it doesn't mean that this gene is more important than this gene. It's just a biological variation. From what else the biological variation came? As I said, it can be naturally because, for example, genes of cytoskeleton, they are expressed much higher level than transcription factor, for example. It also can come from sequencing depth. It can come from different PCR efficiency. So all the technological things contribute to the difference in sequencing, in amount of detected mRNAs. And then you need kind of to make it compatible. All the genes should be compatible with other genes. So we need to transform the data. How do we transform the data? That's called normalization, and typically it contains two parts. It's scaling and transformation. And this thing is needed So you feed to the principal component analysis normalized data, otherwise you will over-represent highly expressed genes. Scaling is a very simple thing. Basically, you have individual number of identified copies in each cell, and then you need kind of find the concentration instead of having absolute numbers. And then data transformation. There is a very good benchmarking study on different transformation methods and how it affects subsequent analysis in our field. And they found that shifted logarithm overperformed all the other methods. But let's try to understand what actually all these methods are. So the shifted logarithm, the simple methods, they are applied per gene in cell. And the complex methods, that's some models, and they are applied per gene. Basically, this requires more computation than this. Here, we need scaling factor. Scaling factor can be just arbitrary, as it's in Syrah, it's 10,000. Or scaling factor can be computed based on your library. So in each individual case. And then you kind of also stabilize variance, having log from it. We end up having these pictures. For example, this how raw data looks. You have lots of zeros in your data. And of course, like mostly statistical tests, they don't like heteroscedasticity, but also they don't like zeros. Shifted logarithm helps to stretch your data, so you have the range. Analytic Pearson residuals mostly are used in case of low expressed genes and for finding rare cell types, because it over-represents lowly expressed genes while you apply this method. Then log with pseudo count from Scrum. It used, I don't think, to be honest, it's used now, but people mention it. And that's coming from Syrah. It's basically a shifted logarithm, but with instead of having pseudo count, instead of having scaled factor computed based on your library, it just takes 10,000. And it looks pretty much as shifted logarithm. This study that I mentioned, they found that simpler methods as a Syrah method, normalization method, or shifted logarithm that is implemented in ScanPy, they overperform all the rest of methods. So let's stick to that, basically. It was a simple and fast part. Key takeaways? Yes, please.
Sorry. So it's even better than SC transform. So Serrat states that their normal basic
yes, ACT transform. Analytic Pearson residuals. ACT transform, yes.
So you're talking about the ACT transform.
I talked it because it's Pearson residuals, but independent study found that it doesn't overperform simpler method. So why would we use ACT transform if it's not good enough?
I think it's making a better VMAP, even VMAPs. Like the graph looks more 3D, but it's just aesthetic. It's not like something.
In my experience, ACT transform overcorrects data, because while you're performing ACT transform, you also include batch, and then you kind of all your data become very much meshed. And we need to be very careful while we correcting any batch, because we also can lose biological differences, and I don't like because I don't control it. In this way, I don't use ACT transform. But that's already like all the flavors of normalization.
Can I ask you a question more practical? Have you ever wondered which normalization should you use? Did it ever matter for you personally?
For me personally, no. For me, no. But also I work with the same cell type, the same data, just then I use all the different subsequent analysis, and for me it was okay. But maybe if you have diverse data and you have different tasks, then we need to think about it, because this paper states that if you have a rare cell type, then go to analytical Pearson residuals. Haven't tried. Then I think that I should feed you. The next question is feature selection, and that's coming from information theory. I think that the paper was published 30 years ago. That while we are having this super highly dimensional, and each gene is a dimension, data, not all of these genes, they are equally informative. Most of them, they are co-expressed and therefore they are co-correlated. And some of them, they are housekeeping. So we don't need all these genes to construct, to do subsequent analysis, because it will be excessive information and lots of noise. If you have tried to do at any point ML, you know that there is always a trade-off in noise and real information, and you don't need to overfeed your data. There is no like real ML right now, but we just don't need to introduce more chaos. It's already enough. Typically, we take 200 up to 2,000 up to 4,000 genes, and we use in different methods. There are different algorithms to find features that are highly variable. And finally, there was a benchmark on that. It was published just a couple of months ago, so it's a really fresh knowledge. And they found that Syrah VST transformation with number of genes 2,000 overperforms the rest, which is very nice. I'm very happy.
Over or under?
How?
Better or worse?
Better. The best. Yeah, and apparently this one, because you don't need to know it. No, it's just technical problem. But basically, Syrah overperforms others. And yeah, for example, ACT transform, not so good. Then the next step before we do PCA, which is a crucial step in this the whole pipeline, we need to do scaling. Scaling is another thing, that's score normalization, and we do it so we stabilize mean and variance. And in Syrah we also can introduce things that we want to regress out. Features that we want to regress out. Sometimes it's helpful. But in my experience, this thing doesn't work very well. For example, if you have proliferating cells, then because proliferating cells, they are so highly co-correlated, it will introduce bias in your PCA and your map. So instead of using this approach, trying to regress out genes that are associated with proliferation, I just removed the whole principal component that has loadings related to proliferation, and it worked much better. So again, just go to the principal component analysis and try to look at the bare genes. Next step is integration. Integration is not always needed. While you're working with in vitro culture, so mice, they are nice and identical, so you're not struggling with it. But if you work with human data, we are super heterogeneous, and you always have some batch. But you still want that T cells all cluster together. Then that's how batch batch effect looks in your data. There are different types of different mathematical models how you address this question, of course, and there is a benchmark on that. Integration and batch correction has a crucial thing that you need still to preserve your biological variance. And that's called bioconservation. And but you want also to correct the batch. So they developed TSLab. They performed this benchmark, and also they developed a package which is called SCIB that gathers lots of different metrics on calculation how well your data is corrected and how well your cell types and biological diversity is preserved. And then they performed benchmarking across these 20 different methods. They found that this overperformed the rest. But you can see that actually they're very similar. So you're interested in this corner, right?
So FastNMN is also solid choice.
Yeah, in how they did it, because it's still there wasn't. They used the same, I think, species, but different tissues. And there was another study that tried to use different species and tried to use all these different methods, trying to understand if the batch is huge, and when species are different, batch is huge, batch effect is huge. And then it's completely different picture. Like, for example, a CVI overperforms, Scanorama doesn't work at all, Seurat goes on top. So it's also all these methods, they work differently on in dependence on the size of your batch effect. Therefore, my advice would be take several of them, and it's actually very convenient because now Seurat 5 has implemented 4 different integration methods in the same command. So you can just try all of them, try to calculate a CIB matrix, or just try to look with your bare eyes at UMAP. If you see that the thing you want to correct are corrected actually.
How is this package called?
SCIB. B, biology, SCIB. We can talk about it later if you wish. Then KTK ways. Visualize your data so you understand. So you understand how bad things are. Yeah, you can use SCANV if you have labels, and consider running several integration methods. Principal component analysis. I'm pretty sure that you all know, and I have to hurry up. So it's very important. Be careful how many principal components you take. That's important. It's affecting the subsequent analysis. Try to rerun analysis several times with different number of principal components. But it's important if you do normal logarithm normalization. If you do SCT normalization, they claim that it's not important. Nonlinear dimensional reduction methods. UMAP or TSNI. You need to be aware that these distances do not reflect actual similarity between cells. So if this one is 2 times less than this one, it doesn't mean that these cell types 2 times more similar than these cell types, because that's not linear. And also, all of them, this one is stochastic. So always set seed. Clustering. Clustering is the thing that is coming after PCA. And clustering is important because what you're trying to find actually cell types in your data. Then first, you just do PCI, so you identify similar cells. Then you're trying for each cell, you're trying to identify some number that you specify of cells that are closest. Then you refine, and then you find some commodities, aggregate network, using Leiden or Leuven algorithm, and identify your clusters. This is very, very subjective step. It depends on analyst, so feel your power in this step, because only you as an analyst identifies actually cell types, so therefore clusters presented. If you can go to lower resolution, you find just Mayer superclusters, like for example T cells and macrophages. And if you want to study something in depth, then you go to higher resolution, you find all the subtypes of your macrophages. So if you are interested in more detailed analysis. Then you need to find cell types. This step is called cell type annotation. There are 2 types of approaches. It's a manual or automated. There are also 2 types of ways to do that. It's top to bottom, bottom to top. Basically, either you know cell types that you expect, you know markers for them, and you visualize it, or you know you have performed some differential expression analysis, you know genes that are expressed, and mark your clusters, and then you try to find using some different databases cell types that correspond to what you see in your real data. We will do that as well during workshop. That's how typically you see result of identification. This you will see in some paper. Different clusters, some genes expressed, and what you want from a gene, from a marker, gene marker, is that it's exclusively expressed in your cell type and not expressed in others, because you want to stain afterwards. There is a benchmark on that, of course, as well. And the good thing that the Syrah-Wilcox test overperformed the rest, and t-test. However, there are different approaches to address this problem, and these two Syrah approaches, they use one against rest. So you test your population against all the rest cells. Also, it's possible to do pairwise, but you need to be aware once you're using one against rest, so you're comparing your target population with all the rest of cells in your sample, and therefore it's sometimes, mostly cell types, some cell type is overrepresented. For example, if you have 90% of macrophages in your sample and you compare, T cells, and then basically if you use one against rest, you compare only macrophages against, only T cells against all the macrophages and not taking into account the rest of cells. So this approach, you just need to be aware what you compare with what, and all the markers that you find, they are subjective. So try to visualize afterwards how well actually it distinguishes your cell type from the rest. The next thing is trying to answer if you have conditions. For example, you treated your sample or your animals with some compound, and then you want to answer a question, what are transcriptional differences in your cell types. That's basically what you are trying to answer. And the conventional way, what you do is that you want to apply again t-test or Wilcox test, trying to identify mean difference in your groups, in particular cell type. The problem is pseudo-replication bias. Although this approach was used all the history of single-cell RNA sequencing, which is around 7 years, this paper has come and changed everything, because they showed that once cells are coming from the same donor, the same animal, they are co-correlated, and therefore you can't use all the Wilcox and t-test because they assume independence of your samples. And your samples, so cells, are not independent if they are coming from the same sample. That's one problem. This leads to inflated type 1 error rates. What else also leads to inflated type 1 error rates is that each cell is treated as an individual sample, and therefore you have super high power, statistical power, because you have thousands of cells on one side, thousands of cells on another side, and you do not fulfill requirement to be independent, and therefore you find lots of differential expressed genes, like thousands of them. To address this question, there were some benchmarking studies, how you basically find way to deal with it. It's very important that you do replicates in your single-cell RNA sequencing study. So having at least 3-4 replicates in one group, so it's sequenced as different library, different batch, and 3-4 samples in your another group. And then the most approved approach now is to do pseudo-bulking and then apply DESEQ, which is a bulk RNA sequencing method, but also it assumes negative binomial distribution. It models correctly type of data that we work with. So that's approach best practices to use. If you have different conditions, you need to have replicates and apply a pseudo-bulk approach to get correct differentially expressed genes. Okay, now sophisticated questions. No, let's go back. Questions, not sophisticated, just questions.
I have one question about the CRISPR component analysis. So I think before in the research materials they included text for Jack square or something like that.
Jack's straw plot.
Yes. And it gave us like exact p-value for each PC. So it was like easy to choose like which one to include, but now I think it was removed. So
Yeah.
do you still use it, or like how do you?
Jack's straw plot, you mean?
Yeah.
Personally, I calculate standard deviation, and then I choose arbitrary threshold, how much variance I want to have covered, and the standard deviation, I also set a threshold. So I accumulate some amount of variance that is explained, and choose this amount of PCs that explain this variance. Zuza? Yes. How?
No.
So I have a question about relation between transformation and integration, because you showed benchmarks for both, but when you want to combine them, so first of all, is it okay to do any type of transformation, like SC transform or algorithm, and then any integration on the data, or are there some constraints, and also the order of steps? So does it only make sense to first transform each batch separately and then integrate? Or maybe in some cases to first integrate and then transform on everything? Do you know where any studies like doing both? And
When you do normalization in Syrah, it takes normalized data. And it's just in pipeline, so anyway you will do that. I'm not sure how it's in ScanPy. Maybe it's also like implemented all together, but it makes a difference which normalization approach you use. Although in Syrah it's just two, SCT or logarithm, and yes, it has difference because SCT overcorrects, but it was not shown anywhere. Just my experience. So typically I do logarithm and then RPCA, because RPCA is less overcorrecting than CCA.
Okay, and when you have different batches, then you do integration like with Harmony or another on the SC transformed data?
Say it again?
So for example, if you use SC transform, or like what you use, and then you do integration on these transformed data?
No, I don't know. I think we can discuss it later.
Okay. Yeah.
I will think about it.
Because I have this problem when you will spatial transcriptomics data, but I think it will be similar.
Yeah, it's going to be similar. I think it's the same techniques. Did you have a question? Okay. So do we continue?
Want to ask, why not use Mann-Whitney since you don't have any dependence or normal distribution, for example?
Say it again.
That you can apply this extension because you don't have any dependence in the sample controls.
Because you don't have. I think I'm
you
deaf.
don't have independence. But why not Mann-Whitney, for example? That doesn't assume independence or normal distribution.
But we do not use Mann-Whitney. We use Wilcox, right? In Syrah, at least.
Okay.
I just wanted to ask that in case if we try out different transformation approaches, doesn't we until we see what we want to see, doesn't we introduce confirmation bias?
Yes, it introduces confirmation bias, but it's always a trade-off, because in science you always have confirmation bias, but also how do you know that you're doing it right? It's very philosophical now discussion, because confirmation bias is something like philosophical. There is no way to measure it, right?
Right.
So yes, that's what we do.
And the other thing that I like to ask is regarding like data use and stuff. Sometimes Monarch 1 is also used as a user stuff, and what is the?
Yes, some people use. I haven't seen any studies that prove Malat 1 as a way to show anything, but some people say that I saw like papers that not focusing on this technology, but doing some biological study, and they found that in their cell type and that cells, Malat1 is overexpressed.
Oh, okay.
So for some cell types, apparently it's relevant. But there is no any comprehensive benchmark if you should include Malat1.
Thank you very much. Thank you.
Can I ask a question?
Yes.
It's fast. it's a good practice to select by the percentage of variance explained and so on. But you're going from 20,000 dimensions to either 20 or 30. Does it actually matter? If you plug just 30 principal components.
But it's amount of chaos that you introduce. You just want to introduce least chaos.
So my question is, if you include 5 more principal components, does it really affect?
I would say yes, because each principal component is loaded with some specific genes. And yes, it affects the consequence. Like not that much maybe, but it also depends on data set. If the data set is highly variable, for example, it's a whole tissue with different cell states, you actually will have lots of PCs that with very interesting loadings, right? And I personally analyze what?
Like a cell cycle, for instance. So
yes.
Because the problem in my particular data is that we have a lot of cells and they're divided, different dividing cells. So I'm afraid that they will cluster based on their cell cycle.
Oh, I think they do. They do.
What?
They do, I think, because.
So then I have to remove the PC that's associated with.
That's what I do typically, because the rest methods, they do not work. It doesn't help to remove cell cycle because it's so strongly co-correlated.
So we just go dimension by dimension from PCA, like you look at, you color the cells by the cell cycle phase, for example, and you look at which PC principal component separates them the most, and you remove. How?
You have loadings per gene, right?
Yes.
So if you have the list of genes, you can see which PC has the highest or lowest or absolute values.
Yes.
But Soyuz already has a method to normalize for cell cycle genes.
Cell cycle are different for different cell types. So these methods do not always work the same at different cell types. For example, for my cells, they do not work. But for maybe highly proliferated cells, they work. I haven't worked with them. So.
When it doesn't work, what do you see?
When it doesn't work?
When it doesn't work for your cells and you try to perform it, what do you see?
Mostly I just see, for example, I used this CRAT cell cycle analysis. I just see that all the cells, they have, it's completely mixture of different cell states and they're not clustered all together. And the difference actually in this score of presence of these cell cycle genes is actually very small. So it doesn't make any biological sense to proceed, I would say. So thank you for all your questions. We can also have a discussion later during workshop. Next, sophisticated questions. So we have identified differentially expressed genes. We have our cell types. What do we do next? First, of course, mostly what people think about, I have thousands or hundreds of differentially expressed genes identified in each cell type, up and down regulated. I want to collapse all of them in some pathways. And that's called enrichment analysis in some analysis and differential genetic enrichment expression analysis. Then another question what you can ask is which transcription factors govern your cells? And this question is answered by constructing gene regulatory networks in ferrin regulons that afterwards you can collapse into models. Then you can ask if this approach is not possible because, for example, your species is not very well annotated or you don't have attack and ChIP-seq data for your species. Then you can just ask which genes are co-expressed. And this is co-expression analysis, for example, is a weight gene set network, VGCNA analysis. There is an application specifically designed for single-cell RNA sequencing. Or you can just construct gene models. Here, this one. Then what you can also do is trying to predict cell fate if you are working with differentiating or de-differentiating data, RNA velocity and pseudo time. And of course, compositional analysis. So pathway enrichment analysis looks like this. You use hypergeometric test to answer which pathways are represented, overrepresented in your data. And of course, this approach is highly dependent on database. Basically, you need to obtain some gene lists with some particular molecular functions. You can use mSeqDB, you can use Reactome, KEGG pathway, Panther. There are many of them. And there are different tools to do it online. So, yeah. And gene set enrichment analysis is more powerful because it allows you to have background genes and to have up and down regulated. Yes, and the thing, fun and cool thing that looks nice in your papers is trying to visualize any gene set that you want to visualize using add model score Seurat function or UCell. Basically, it's like you have a gene set, I don't know, let's say snail transcription factor and or TGF-beta reaction response. And then you just visualize this gene set, how it's active in your cell type, in your cell types, in your data set. Compositional analysis. That's a very important question, and so far not so many people, not so many analysts used compositional analysis, although it's a double-sided thing, because while you are clustering your data, you end up in different clusters and if difference between cells is big enough. And if the cells are not that different, then they will end up in the same cluster, and while you are doing comparison between one condition and another condition in this particular cell type, you will end up basically having some DEX, and if your effect of your treatment is large enough, then it will produce maybe even new cell type. But you won't compare it with within the same cell type because it's, the difference is so big that you ended up having another cell type. So compositional analysis is always must have to do, because then you will find out differences in cell abundance and new clusters. There are multiple approaches to do that. It's we will use two, CACO and DASEC. Also, these use Bayesian methods, Bayesian inference, but of course you need to have some premises to use them, and otherwise it doesn't make any sense. And Milo-R, which is based on connectivity and neighborhoods.
We will use the DSEC with the ASEC with dash or without?
I think with dash. It's the same actually. I just typed it twice. Gene regulatory networks. It's actually a very cool thing, mostly used as a scenic. You need to have some preliminary data, but it's databases that are available. And basically then there are steps how you do that. You find co-expressed genes, then you know motifs for them, and you're trying to predict which transcription factor govern these co-expressed models. Then you're trying to use AUSU to calculate area under the curve, trying to understand if this regulon, so group of co-expressed genes under control of transcription factor, is represented, how it's represented in each individual cell. So basically this approach helps you to identify regulatory mechanisms, although it doesn't make any sense to do if you can't validate it. So all these sophisticated things, they have to be validated in some wet lab methods. The next is pseudotime, and pseudotime is an approach that helps us to answer a question, what is the differentiation trajectory and about consequences in your data. The approach comes and stems from the idea that differentiation process is asynchronous. So while you have snapshot of your data, you have all the possible stages at the same time. So you have all these cells, and then you are just trying to predict how they align. This is basically based on similarity of cells and the gradual shift in transcriptional profile. There are some assumptions in this type of analysis. Basically, you have to fulfill the requirement by yourself that each stage of the process is present in your data. It can't infer any stage. Then you have to be sure that it's a continuous shift in the expression profile in cell states. from a root population that you should know to end population that it's desired that you know it, and the changes in gene expression are steady and gradual. So all these three assumptions basically typically are fulfilled when you work with developmental data, and when you work with pathological processes, it's rarely fulfilled. But I mean, we have to be conscious about it. And the thing is that trajectory can be forced on any data set without any biological meaning. You can just take any data set, calculate a slingshot, monocle, and you will find some trajectory. It doesn't mean that this trajectory really exists. There is a paper from 2025 that addresses this problem, and of course they developed their own tool that overperforms others. That's a typical thing. But typically there is a thing called double dipping, and double dipping is a problem in all single-cell RNA-seq data, because after PCA you do your map and you construct some structure of your data. And this is done basically on loadings of PCA. So how different genes variably expressed in your cells. And then you again go back and try to test differential expression along this trajectory. But your data set has already been based on your PCs. So it doesn't make any sense to, well, it makes a little bit of sense, but not that much. If you are trying to understand what genes are different across this trajectory, because this trajectory was constructed based on PCs, if it makes sense what I'm saying. So double dipping problem is not addressed so far. It's just how these things are. Basically now all the majority of trajectory methods that you can find, most popular is slingshot, monocle, they basically you just you draw a line. You need to specify a route, you draw a line, and then you just have these numbers that you can correlate with genes or whatever. It's just a vector, basically. But interesting thing comes when time series data comes into play and you have time series, because then you actually can model something. There are tools for that, SuperTime, ChronoCell, LEAP. LEAP allows to include transcription factor information. You can also try to model different methods, assume different behavior of biological cell types, because this model were created based on different biological data, and then they try to infer this behavior on your data set. Next method is RNA velocity. I think it's one of the most fun method, but although it's widely misused. Because you are allowed to use RNA velocity data only if the process that you study has a time scale is a half-life of mRNA. Because you are allowed to, there are some assumptions how you use RNA velocity. First of all, the assumption is that accumulation of, well, first mechanism, how it worked. mRNA has two stages, basically. It's spliced and unspliced. Unspliced come first because it's so-called unmature RNA, and then RNA matures. And then having these sequences spliced and unspliced, and you know that first comes this and then this, you kind of have a vector direction of for your cells in time, but in your single point, time point snapshot. Therefore, accumulation of unspliced, then degradation, this circle is time dependent, and you can infer real time based on your snapshot data. But although you need to check that your curve of accumulation of unspliced and spliced counts corresponds to model that of tool that you use, because there are different tools and they have different models, and they specify which model they infer. So you need to check, they provide some plots. You need to compare what gene expression you have and how they assume that you have, because if it doesn't fulfill the requirement, the tool will work poorly. I think that's all. It's just a pipeline in Syrah. This is Syrah object. Always remember the golden rule of bioinformatics. Garbage in, garbage out. And I gathered some useful links. This lecture will be available afterwards. So, yes. Now questions, and the workshop will be in 5 minutes.